KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Hi students, now in this video we are discussing cell wall information. So in previous video we have seen information of cell membrane. So we have learned a very extensive study, exclusive study on cell membrane in previous video. So in this video, so let us discuss the complete information of a cell wall and the chemical components which are involved in the cell wall. Entire detailed information I will give you, and after completion of this particular information, we will be concentrated on the endoplasmic or endomembrane system. So here we will be finding of presence of endomembrane system. So means in this complete video, cell wall and endomembrane system. So in endomembrane system, so there are different types of cell organelles are there. So each and every point, so what is information is there in our NCT textbook, every point, so I will give you, so we will discuss. Now let us start to discuss cell wall. Now cell wall is a non-living rigid structure. This is very important point. Suppose if you take the cell wall, so this particular cell wall is a non-living rigid structure, forms an outer covering layer for the plasma membrane. So thereby here, first of all, actually, so who discovered, sir, this one dead form of living cell? Who discovered means Robert Hooke. Who is that? Robert Hooke. Actually that particular point, so in a NEET only I have to give, but take this particular point, don't forget. First of all, dead cell or dead tissue, cell wall, dead tissue or the living rigid structure, dead material, or dead means non-living only, so dead material which is discovered or which was discovered by Robert Hooke. Okay, Robert Hooke. So thereby what it will do, what is the function of this particular cell wall means, the cell wall is a non-living rigid structure, forms an outer covering. Definitely forms an outer covering for what? Plasma membrane. So means definitely plasma membrane is covered by, plasma membrane is covered by, definitely by cell wall. So where we can see the presence of this particular cell wall, definitely we can see in fungal members and plants. Okay, so definitely, so in a fungal members and plants, definitely we can see the presence of a cell wall and which covers the plasma membrane. Okay, so that particular point you try to save in your mind. Now the cell wall not only gives the shape to the cell, definitely which provides the shape to the particular cell and protects the cell from the mechanical damage and infection. Point you understanding, Munadi in anatomy we learn, epidermis is outermost protecting layer, likewise we learn, in anatomy, particular cell point of view, outermost protecting layer is cell wall. This particular cell wall not only gives a shape to the cell and also provides or also protects the cell from the mechanical damage and infection. Definitely it provides the shape. Cell wall. So it provides the shape to the cell, not only providing of a shape, definitely it involves in providing of it involves from the protection. So protection from the mechanical damage and infection. Next one, it also helps in cell to cell interaction. Which one? Cell wall. It also helps in cell to cell interaction and provides barrier to undesirable macromolecules. Or barrier means like a gate madri, so that provides a barrier to the undesirable macromolecules. So here definitely, so this particular protecting layer is a cell wall which provides the cell to cell interaction, which helps in cell to cell interaction and provides a barrier. Definitely that is only a gate like structure in the cell to what? Undesirable macromolecules. So suppose if unwanted macromolecules or undesirable macromolecules are there, means definitely this particular cell wall object them or will not give permission to them to enter into the cell. Okay, next one, suppose if you take, there are different types of cellular like organisms are there, no? So now let us see. So in those kind of like cell organelle or in those kind of organisms, no, how the cell wall is made up of. Now let us see one by one. So algal members, algae have cell wall made up of. Definitely if you see algal members, no, so what we can see, algal members, if you see their cell wall is there, that particular cell wall is made up of cellulose, galactans and mannans. Important, NEET examination point of view important. So once you see algal members have a cell wall made up of cellulose, galactans and mannans and minerals like calcium carbonate. So like while in other plants, okay, while in other plants, so for example if you see there, no, consist of cellulose, hemicellulose, pectin and proteins. So cellulose, hemicellulose, pectin, proteins, likewise there are different types of chemical components are present in another plants. But in algal members point of view, definitely cell wall is made up of cellulose, galactans and mannans. This definitely in a slip test examinations I will give you. So nalla save pannikonga. Definitely algal cell wall is made up of cellulose, galactans, mannans. Line wise. Next one, along with these kind of chemicals, minerals also present. Minerals like, what is the name? Calcium carbonate. Calcium carbonate chemical compounds also are present. Suppose if you take other plants, definitely they consist of cellulose, hemicellulose, pectin. Not only these three things, proteins also, or there are different types of proteins also are present. Next one, the cell wall of a young plant cell. From here onwards, important. These points also important, but the importance of the concept is going to be increased. Okay, now let us see the plant cell. The plant cell of young plant is a primary cell wall. So here, if you take particular cell wall, in a young plant. So if you take in young plant, definitely we will observe the young cells. So there what we can find out means, so we will find out the presence of a primary cell wall. So primary cell wall is capable of growth. Don't forget. So until completion of your biology related studies, you have to remember this particular point. So means, suppose if you take particular cell wall, so this particular, once you listen carefully, what is information? Cell wall, our knowledge point of view, we can be divided into two types. So one we are learning here, what is a primary cell wall? Suppose one is a primary cell wall, means definitely second one will be secondary cell wall. Point you understood? Easy da. Cell wall, our knowledge point of view, let us divide it into two types. One is primary and one more is secondary. If primary cell wall is capable of growth, if particularly this one is capable of growth, means the second one doesn't have point you understood. So thereby here what is the point? In the cell wall of young plants, the primary cell wall is capable of growth, which gradually diminishes as the cell matures and the secondary cell wall is formed in inner side of the cell. Once you see, so means what is the capability of the primary cell wall is there now, that particular capability is diminishes. So we have seen diminishes means gradually reduce that capability, and when when the particular cell is going to be matured, as the cell matures. Next one cell wall, next type is a secondary cell wall. It is formed inner side of the cell. Inner side of the cell means, sir, end side we have to observe means, in brackets we have written once, you see, inner side of the cell means towards the cell membrane. Once here one small rough diagram I have drawn, but clear cut information I will give you there once you see here what is the point. No, here small diagram is there, no, chinna or rough diagram is there, no. Once I will zoom it. Once you see here, yellow color structure, outermost protecting layer is there, no, or yellow structure is there, no, this is middle lamellum, and ulle colors not appearing that much clearly, but ulle pink color is there, no. First one yellow layer, middle lamellum, ulle middle lamellum ulle we are observing the presence of cell wall. There cell wall, our knowledge point of view, we have been divided into two types. No, one is a primary and one more is a secondary. Primary one, that is situated towards the middle lamellum. Later secondary cell wall, which is situated towards the plasma membrane. Dots you are observing, no, the dots are nothing but cell membrane or plasma membrane. Simply generally I'll write it as a CM or PM. CM means cell membrane or PM means plasma membrane. Easy only, no. Inner layer, what is the layer we are finding? Dot structures are there, no. Adi yarana plasma membrane or cell membrane. Towards the plasma membrane in cell wall, no, towards this plasma membrane. So we will find the secondary cell wall, and that side, no, primary cell wall. Next to that side we will be observing middle lamellum. Okay, so when you will be here, so this particular diagram I will give you by one more time, but here what is the point? No, so to give you that particular information I have zoomed it. Okay, I zoomed it, but here listen carefully, so it is this particular information also is important. Okay, I hope you understood that particular diagrammatical explanation there. Okay, now here what is the point now I am going to give you is, so here if you see the structure of the cell wall, so that can be divided into two types of for our convenience to study. What is like a situation means, this particular primary cell wall, it is present towards the middle lamellum, and secondary cell wall which is situated towards the cell membrane. So thereby I hope you understood this one. So but suppose if you have any doubt, no, once you practice the particular cell, practice the cell, so thereby very clearly you will understand. So thereby here yellow layer we have drawn, no sir, yellow layer why sir you have drawn, what is the relation of this one with the cell wall means, in plants, no, so all cells are like comes together and form a tissue, no. Cells are there. Suppose when this particular middle lamellum is absent, no, cell walls or the cells cannot be situated as a tissue, or we could not see the presence of tissue. Why? Because actually what is the point now I am going to give you is, wall is there, no, construction, wall we will construct, no, at the time bricks we will keep there, and between the bricks, no, we will put cement. Try to understand clearly. Normal example. So bricks we will be keeping there, and beside to the bricks or between the bricks, no, we will be keeping of cement. So likewise here also between the cells or to hold the cells, so between the cells, no, so God has arranged which one, middle lamellum. Middle lamellum. Presence of middle lamellum, what it will give, sir, means when the particular middle lamellum is present, no, so all cells comes together and form a tissue. So thereby here this particular middle lamellum, actually here information not there, in a NEET examination point of view I will give you. So this particular middle lamellum is acting as cement like substance to hold all cells together. Cells, when all these cells are present together, no, that time only we can call a group of cells as a tissue, isn't it? So thereby here for the formation of tissue, main important role played by middle lamellum. point you understood. So that's why this particular middle lamella is outer one. Okay, next one ulle cell wall. Suppose a cell wall nalli if you are dividing into two types means, so definitely primary one and a secondary one. Okay, primary one which is nearby or which is very close to the what middle lamella. Next one, a secondary cell wall also is present, that one very close to the plasma membrane or cell membrane. Okay, so thereby once you see order middle lamellum, primary cell wall and ulle coming from outside to inside one more time you see there middle lamella will be there, next one primary cell wall will be there, next one ulle secondary cell wall will be there, next one completely ulle plasma membrane or cell membrane we will observe. So here this particular information also try to listen carefully. Next one, the middle lamellum layer. Middle lamella is a layer mainly which is made up of calcium pectate which holds or glues. You see that? So definitely the presence of a middle lamellum or this particular middle lamellum layer is composed of which one? Calcium pectates. And these calcium pectates which holds or glues, glue mean gum we will be simply calling now, so we will be calling by one more name glue, glue. Okay, and the different neighboring cells. So what it will holds means that holds the different neighboring cells together. I hope now you understood this particular point. So thereby middle lamella definitely acting as a gum and it holds all neighboring cells together. So thereby then only one particular formation of tissue we can observe. Now let me move forward. That is the cell wall and the middle lamellum may be traversed by plasmodesmata connections. So thereby middle cell wall is there and middle lamellum is there. So definitely between these structures, one important bridge-like structures. I think so in anatomy starting while explaining about the meristematic cells, I have given information regarding plasmodesmata connections. Plasmodesmata connections bridge-like structures between the cells. Now only we are learning all cells are situated together because of the presence of middle lamella we are saying. All cells are like a group or a tissue madri irukku. Suppose when water molecules and minerals movement is there, suppose between the cells there is no connection means how water molecules and minerals and food materials will transport the one place to another place, no chance, that's why here plasmodesmata connections, so some kind of bridge like structures. Okay, so plasmodesmata connections are present between the cells and which connects the, once you see, which connects the cytoplasm of neighboring cells, means one cytoplasm of one particular cell is connected to the another cell's cytoplasm. So likewise, all cells cytoplasm is connected together because of the presence of plasmodesmata connections. I hope you understood this one. So means because of one land irikama, suppose on the side land, in the side of land, suppose river is there means we will construct the bridge. So likewise here also on the side cell, in the side cell, rendume connected together because of the presence of plasmodesmata. So next one. So that is a small information regarding cell wall. So now let me explain the information of endomembrane system. So endomembrane system there, while each of the membranous organelles is distinct in terms of structure and function. Means one more time you read it, while each of the membranous organelles, so each and every cell organelle having distinct information in terms of its structure and function. Means suppose if you take each and every cell organelle, so that has its own, or that particular cell organelle has its own structure and it has its own function, isn't it? So thereby the structure point of view and the functions point of view, definitely each and every cell organelle having or maintaining distinct nature. So in terms of structure as well as function, means what is a simple point now I am going to give you is LMA cell organelles structurally functionally differ. So structurally difference will be there as well as a functional point of view also difference will be there. So that is only we are learning that point. Next one, many of these are considered as together. Now not only we are learning, there are different types of cell organelles over there. All these cell organelles are structurally and functionally distinct. Likewise separation is there. Likewise we are seeing no. Ipo paringa, what is the point? One more, many of these are considered together. Inge many of the cell organelles we are considering together as endomembrane system. Means there are different types of cell organelles are there, and many of them we are considering under endomembrane system. Sir, ipo ellame cell organelles are there, then why sir we are calling very few cell organelles as endomembrane system, and why others or other kind of cell organelles why we are not considering as endomembrane system? What is the difference, sir? Means we will learn. So now that point only we will learn. Now once you see, many of these are all considered together as endomembrane system. So because their functions are coordinated. So means what are the cell organelles of functions are coordinated? No, all such kind of cell organelles we will be considering as endomembrane system. Suppose if the functions of cell organelles are coordinated, so such kind of cell organelles altogether we will be considering as endomembrane. We will be considering as endomembrane system. Endomembrane system. So now let us study one by one what are the cell organelles which are present in the or which are comes under endomembrane system. Now let us see. Okay, so endomembrane system. So endomembrane system consideration, once you if you see there now, so endomembrane system, so that particular endomembrane system includes endomembrane system include endoplasmic reticulum. First one is endoplasmic reticulum. Second one is Golgi complex. Idi 4 cell organelles are there under endomembrane system. 4 on the spot you have to by heart. Okay, so first of all endoplasmic reticulum, second one is Golgi complex, third one is lysosomes, and fourth one is vacuoles. Now total how many are there? One is endoplasmic reticulum, second one Golgi complex, third one lysosomes, and fourth one vacuoles. So totally 4 cell organelles which are included under endomembrane system. Why these 4 cell organelles only we have taken? Why? Because these 4 cell organelles functions only coordinated. In the side we learn now functions are all functions of the cell organelles. So suppose if coordinated means all these cell organelles we can be taken under endomembrane system. I hope we understood. Next one, now since here once you see, since the functions of the mitochondria, chloroplast and peroxisomes are not coordinated, or the functions are all not coordinated with the above components. With above components means we see no 4 different types of cell organelles. Means here 4 functions or 4 cell organelles functions only coordinated, but remaining cell organelles if you observe there, no, so mitochondria is there, chloroplast is there, peroxisomes are there, still so many number of cell organelles are there. Then why these cell organelles we didn't take under endomembrane systems are? Means once you see these cell organelles functions are not coordinated. That is the main important difference. Next one, these are all not considered because of this. When these functions are not coordinated, no, because of this particular statement, we are not considering these mitochondria, chloroplast, peroxisomes, all these cell organelles. We are not considering, or we are not considering the part of endomembrane system. Easy only now. So nalla save pannikonga. Next one, so first one, so means in endomembrane system, so first one is there, no, endoplasmic reticulum. That one let us start to discuss. So first endoplasmic reticulum let us start, later Golgi complex, later lysosomes, vacuoles, likewise. So let us learn one by one. After completion of these 4 cell organelles, mitochondria, chloroplast, next one microbodies, every information, each and every point I will give you. Right. Now first one, endoplasmic reticulum. So simply we can take this one as ER, endoplasmic reticulum. So this particular endoplasmic reticulum for the first time discovered by K.R. Porter. Porter, scientist name K.R. Porter. If you see this particular structure, no, so that particular structure that gives the information. So where this particular endoplasmic reticulum is situated. Once you see here, nuclear, this NM is there, no, NM, because of the lack of space, no. So simple clues I have written. NM means nuclear membrane. This outer nuclear membrane, exactly at light here NM we are finding, no, this outer nuclear membrane, and one more this line is there, no, this inner nuclear membrane. So thereby we are learning one point. In a nucleus also, in a nucleus how many layers are present? So one is outer layer and one more is inner layer. So thereby two layers are there, and between these layers, no, here we are finding zero structures. Once you see there, motte spherical shaped structures we are observing, no, so like a zero madri. So these structures we will be taking as nuclear pore na hole. So this particular thing. Next one, endoplasmic reticulum here is there, no, that one is situated between, listen examinations point of view important, which is situated between outer nuclear membrane, or which is situated between correct da? So it is situated between the outer nuclear membrane to plasma membrane. Plasma membrane or cell membrane we are taking, no. So thereby, so what this particular endomembrane system, which is situated between outer nuclear membrane to up to plasma membrane or cell membrane. Okay, so that is diagrammatical information. So still we will depend on this particular diagram for learning of this information. So once let us learn the information, later let us explain the diagram. Now you see so now electronic microscope studies, EM means electronic microscope, EM studies. So according to the like which one, electronic microscope studies of eukaryotic cells reveals the presence of a network. Reveals the presence of a network or a reticulum of tiny tubular structures. Network or reticulum of tiny tubular structures are scattered in the cytoplasm. So what are the tiny tubular structures? Tiny means small, T-I-N-Y, tiny means small. Small tubular structures are scattered in the cytoplasm, like how? Network like or reticulum. Network or a reticulum type, where these are all like scattered in the cytoplasm. Now we learn from, so here we can continue. So all these small tubular structures are scattered in the cytoplasm. From we can extend, no. So from outer nuclear membrane to plasma membrane is referred as endoplasmic reticulum. Point you understood, easy only, no. Next one, this particular endoplasmic reticulum, hence endoplasmic reticulum divided the cytoplasm into two types. So here this particular endoplasmic reticulum divides the intracellular space, intracellular space, intracellular place. Intracellular space means, what is the meaning? Intra means within the cell. Intra means within the cell, inter means between the cell. Okay. Intracellular space means the gap which is present within the cell is divided into two compartments or two distinct compartments. One time a diagram paringa. Here is the endoplasmic reticulum. Pink color lines are there, no? This is endoplasmic reticulum. When this endoplasmic reticulum moving from the nuclear membrane to plasma membrane, so actually in absence of these two lines, no, here is the LMA cytoplasm dana. Here nucleus is there, and here entire cytoplasm matrix like a fluid we see, no. So here LMA we will be taking as cytoplasm, and this plasma membrane on the side, no, cell wall we will observe. When this particular endoplasmic reticulum moving from the outer nuclear membrane to plasma membrane, so here this particular cytoplasm is divided into two compartments. So what are those? One is luminal place. What is a place which is present inside of the or inside endoplasmic reticulum? We will be taking as luminal. That's why luminal I have written here. So ఇది luminal and வெளித இருக்கணும். So what is the empty space or what is the space which is present within the endoplasmic reticulum, we will be taking as luminal, and what is the space which is present. So next on the side, outside of the or outside of this particular or at the cytoplasm location, so we will be taking as extraluminal. Extra means outside, extra means outside. So here extraluminal, luminal means within the endoplasmic reticulum, extraluminal means so that is situated outside of the lumen, outside of the luminal. So thereby simple thing, so endoplasmic reticulum so divided the intracellular space into two compartments. So simply one is luminal and one more is extraluminal. Extraluminal means the place is nothing but cytoplasm compartment, luminal means within the endoplasmic reticulum. So diagrammatically you can understand, so here it is cytoplasm then, and here also cytoplasm is there, but here empty space is there, no, or the luminal place is within the endoplasmic reticulum, is outside of the endoplasmic reticulum, extraluminal space. Next one, endoplasmic reticulum often shows, endoplasmic reticulum often shows ribosomes. So I will conclude with soon. So now you see there, so what is the endoplasmic reticulum consists of ribosomes, no? So because of the ribosomes, so the outer surface of the endoplasmic reticulum seems to be rough. So that particular endoplasmic reticulum we will be taking as rough endoplasmic reticulum. Simple, here endoplasm, once again come on to the diagram. So from here to there endoplasmic reticulum. When this particular endoplasmic reticulum consists of ribosomes, once you clearly try to observe there, on pink color lines we are observing chinna white structures, no? These structures are nothing but ribosomes. So now endoplasmic reticulum, because of the presence of ribosomes, that particular endoplasmic reticulum we are calling as that particular endoplasmic reticulum we are calling as rough endoplasmic reticulum. Why? Only because of the presence of ribosomes on the surface of the endoplasmic reticulum. Okay, suppose if here ribosomes are absent, in absence of the ribosomes they appear smooth and are called smooth endoplasmic reticulum, RER. So smooth endoplasmic reticulum, SER, and rough endoplasmic reticulum, RER. Rough endoplasmic reticulum, when we will call, when endoplasmic reticulum comprises or consists of ribosomes, RER forms. Smooth endoplasmic reticulum means here are no ribosomes, there is no presence of ribosomes on the endoplasmic reticulum. Understood? So simple only. So suppose if on the surface of endoplasmic reticulum ribosomes are present means rough endoplasmic reticulum form. Suppose on the endoplasmic reticulum ribosomes are absent means smooth endoplasmic reticulum form. Simple. Next one, once you see rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis. So because of the presence of ribosomes, definitely, so here once in prokaryotes we see, no, in polyribosomes. So there also we see protein ribosomes main important function is protein synthesis we see. Likewise, so here also on endoplasmic reticulum ribosomes are there now. These ribosomes are also involved in protein synthesis, and one more is secretion and protein synthesis. So definitely will be under the control of RER, rough endoplasmic reticulum. Next one, they are extensive and continuous with the outer membrane of the nucleus. So thereby this rough endoplasmic reticulum diagrammatically, this rough endoplasmic reticulum situated very close to the outer nuclear membrane. Next one, smooth endoplasmic reticulum. So smooth endoplasmic reticulum is major site for the synthesis of lipids. Examinations point of view important. Smooth endoplasmic reticulum major site, or this particular SER is a major site for the synthesis of lipids. Means protein synthesis will be under the control of rough endoplasmic reticulum, lipid synthesis will be under the control of smooth endoplasmic reticulum. Next one, in animals cells lipid like steroidal hormones. In animals cells lipid like steroid hormones are all also synthesized. So means here within the cells, you know, or within the where in animal cells, so lipids like steroidal hormones, steroids or simply steroidal hormones not required, steroids you try to remember. So means in animals this particular smooth endoplasmic reticulum helps in the synthesis of steroids. In animals, no, smooth endoplasmic reticulum helps in synthesis of steroids, and in plants, no, definitely helps in synthesis of lipids. So this is a small information, so I can give related to the endoplasmic reticulum. Okay, so these are all major important points. So in the next video definitely I will come with next cellular organelles of endomembrane system. So listen carefully. Suppose if you are unable to understand, so watch this particular video one more time, so definitely you will understand. Thank you and be safe at home.